Good morning, I am Madhurima Datta, Zoology and Biology Educational Content Creator. Today's content for B.Sc Zoology Honours, M.Sc Zoology, CUET PG and also CSIRNET Life Sciences students. Paper Non-Coordinates or Diversity of Life Form of CSIRNET Life Sciences and Unit PORIFERA and topic is Canal System of PORIFERA. For playlist of B.Sc Zoology Honours, M.Sc and CSIRNET Life Sciences, please subscribe my YouTube channel Biogenesis by Madhurima. So, canal system of sponges. Sponges feed primarily by collecting suspended particles from water pumped through internal canal systems. Water enters canals through a multitude of teeny incurrent pores in the outer layer of the cells called pinacoderm. This is an important question. What is a pinacoderm for marks 2 or marks 1? Next, canal system description. Incurrent pores called dermal ostia have an average diameter of 50 micrometer. Inside the body, water is directed past the choanocytes, where food particles are collected on the choanocyte collar. The collar is made up of many finger-like projections called microvilli, spaced about 0.1 micrometer apart. The use of the collar as a filter is one form of suspension feeding. Sponges non-selectively consume food particles sized between 0.5 micrometer to 50 micrometer. The smallest particle, accounting for about 80% of the particulate organic carbon, are taken into choanocytes by phagocytosis. Choanocytes may acquire protein molecules by pinacocytes. Two other cell types, that is pinacocytes and archaeocytes, play a role in sponge feeding. The capture of food depends on the movement of water through the body. There are three main designs for the sponge body differing in the placement of the choanocytes. In the simplest asconoid system, the choanocytes lie in a large chamber called the spongocele. In the cyconoid system, the choanocytes lie in canals, and in the leuconoid system, the choanocytes lie in the distinct chamber. Types of canal system are asconoid type. Asconoid sponges have the simplest organization. Water is drawn into the sponge through microscopic dermal pores by the beating of large number of the flagella of the choanocytes. These choanocytes line the internal cavity known as the spongocele. As the choanocytes filter the water and extract food particles from it, used water is expelled through a single large osculum. The design has distinct limitation because choanocytes line the spongocele and can collect food only from water directly adjacent to the spongocele wall. Were the spongocele to be large, most of the water and food in its central cavity would be inaccessible to choanocytes. Thus, asconoid sponges are small and tube-shaped. As an example, examine Leucosolenia, where slender tubular individual grown in groups attached by a common stolon or stem to object in shallow seawater. Next is cyconoids type canal system. Cyconoid sponges look somewhat like larger edition of asconoids. They have a tubular body and single osculum, but the body wall, which is really the spongocele lining, is thicker and more complex than that of asconoids. The lining has been folded outward to make choanocyte-lined canals. Folding the body wall into canals increases the surface area of the wall and thus increases the surface area covered by choanocytes. The canals are of small diameter compared with asconoid spongocele, so most of the water in a canal is accessible to choanocytes. Water enters the cyconoid body through dermal ostia that lead into incurrent canals. Water enters the cyconoid body through dermal ostia that leads into incurrent canal. It then filters through tiny opening or prosopiles into the radial canals. The beating of the choanocytes flagella forces the used water through internal pores or apophyles into the spongocele. Next, food captures does not occur in the cyconoid spongocele, so it is lined with epithelial type cells rather than the flagellated cells present in asconoids. After the used water reaches the spongocele, it exits the body through an osculum. Next, leuconoids type of canal system. Leuconoid organization is the most complex of the sponge types and permits and increases in sponge size. In the leuconoid design, the surface area of the food collecting regions with choanocytes is greatly increased. Here, the choanocytes line the walls of small chambers where they can effectively filter all the water present. Leuconia, that is leucocyte, for example, is a small leuconoid sponge about 10 centimeters tall and 1 centimeter in diameter. It is estimated that water enters through some 81,000 incurrent canals at a velocity of 0.1 centimeter per second in each canal. Because water passes into flagellated chamber with a greater cross-sectional area than the entry canal water flow through the chamber slows to 0.001 centimeter per second. Such a flow rate allows sample opportunity for food capture by choanocytes. Leuconia has more than 2 million flagellated chambers where food collections occurs. In Leuconia, all water is expelled through a single osculum at a velocity of 8.5 centimeter per second. A jet force of carrying used water and waits for enough from the sponges to avoid the refiltering. Some large sponges can filter 1500 liters of water a day, but unlike leuconia, most leuconoids form large masses with the numerous oscula. Leuconoid bodies account for most species within class Calcarea and are the most common type in other classes. Today's topic has ended.